Hello, we're glad you joined us for this live webinar, Integration of Technology, Process and Quality Control, Protecting Complete Sample Chain of Custody. I'm Alexis Cross of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots and brought to you by Brooks Life Sciences. For more information on our sponsor, please visit brookslifesciences.com. Now, let's get started. You can submit questions to the speakers during the presentation while they're fresh in your mind. To do so, simply type them into the drop-down box located on the far left of your screen labeled Ask a Question, and click on the Send button. We'll answer as many questions as we have time for at the end of the presentation. If you experience technical problems seeing or hearing the presentation, just click on the Support tab found at the top right of the presentation window, or report your problem by typing into the Ask a Question box located on the far left of your screen. This is an educational webinar and thus offers free continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process of obtaining your credit. Lastly, at the end of the presentation, you will receive a survey, which we kindly ask you to complete. Your answers and suggestions will help improve our webinar program and are greatly appreciated. I now present today's speakers, Andrew Brooks, Faye Betsu, Shawn Levy, and Brad Hamilton. For a complete biography on our speakers, please visit the Biography tab at the top of your screen. Dr. Brooks, please begin the presentation.
Great, thank you very much. Welcome everybody. Thank you for joining us today. I'm Andy Brooks. I'm the Chief Operating Officer of RUCDR Infinite Biologics and the Chief Scientific Officer for Brooks Life Sciences. My day-to-day work is around biobanking services, a lot like all of you that are listening in today, with a focus on understanding sample quality control. And today's presentation and discussion is really about managing the complete digital and physical chain of custody for the biological samples that we're all responsible for. I'm really delighted and honored to have a tremendous panel that can participate in this discussion. And everyone, as you'll learn from the brief presentations that are going to be made, comes at the field of biobanking and sample quality and data quality from a slightly different perspective. We have Faye Betsu joining us, who's the Chief Scientific Officer of the IBBL, the Integrated Biobank of Luxembourg. Faye is a world-renowned biobanker and molecular biologist that has really focused on the quality around biosamples and has set industry standards truly in the technologies and the approaches that she's employed in understanding sample quality. We have Shawn Levy, who is at the HudsonAlpha Institute. Shawn is a world leader in sequencing-based services and technologies. He supports several thousand projects around the world. And Shawn's perspective really relates to the use and quality of data, not just data integrity, but the quality of data that is a function of the biological samples that we collect and process and present. So understanding the role of quality control and how it relates to the data that we all work with once we analyze these samples that are being collected is a tremendous perspective. And our last speaker and panelist is Brad Hamilton, who is the co-founder and Chief Scientific Officer of Lifevault Bio. Brad's company has a very interesting perspective in the direct-to-consumer market in being able to respond and react to the business and science of personal biobanking, the collection of samples by individuals that want to store biomaterials that can relate to their own healthcare in the future. And as you can imagine, the quality of samples associated with that kind of activity is critical. So today's agenda, we are going to be doing a roundtable discussion, and some of the discussion points that we're going to cover through a series of short presentations, then followed by a Q&A, are a discussion on biobank standards and how they impact sample chain of custody, sample management, the processing quality, control tools that enable harmonization, and how we can all work with samples in a manner that allows us not only to compare data, but compare different analyses, as well as collaborative biobank efforts in precision medicine and what sample quality means to personal biobanking as well as institutional biobanking. Managing the sample life cycle, which is something that Brooks Life Sciences has built their life sciences pipeline around and that we employ, is really about harmonizing biobank essentials, all the way from the very early times of planning your studies, facilitating sample collection, transporting that sample at the right temperature with the right timelines, the processing of that sample, and then the ultimate storage and distribution of those samples. So what's really critical is to understand how all of quality control fits into this process. And I think one thing you'll find from all of the speakers and all of the things that we do as a community is that science and technology is the true driver of innovation. By integrating sample collection, processing, integrating sample collection and processing with an eye on the types of analyses that we're doing, we can best protect those samples and ensure that we're going to have the highest quality data. To do that, we have to understand what the future of sample analysis is, the technologies that have yet to be discovered, the applications that have yet to be investigated. But we need to create sample quality control metrics to standardize across all collections so that we're all operating from a similar playing field as best we can. There are many opportunities for adverse sample and adverse data collection events to happen. It can be at the time of sample collection. It can certainly be at the time of processing of that sample, and as well as the time of storage of that sample. All of these processes need to be regulated. They need to be harmonized across different programs so that we can expect the same quality irrespective of where we get our samples of. The way I personally look at it is breaking these things down into what I call the functional essentials, how we maximize biological resources. All of these slides will be available, so we're not going to spend too much time on any one slide, but the key points here is how we maximally use each primary sample, making sure that the chemistries and technologies we use for processing are maximally efficient. As the samples that we deal with are precious and limiting, we want to maximize the processing of them. Defining appropriate storage formats, using the proper tubes, the right automation, so that we handle the samples correctly post-processing. Creating renewable resources, whether it's nucleic acid amplification or cell line establishment, as Brad will speak to later on. And then also establishing the right kind of governance, the appropriate distribution guidelines, so that when people request samples, we provide what they need to be successful and don't put the rest of the collection at jeopardy. And hopefully the theme that you will hear throughout the other speakers is how we need to integrate quality control across all of what we do. We must implement technologies to evaluate samples as a part of that omics central dogma. You'll hear Shawn speak to the kinds of QC that's done to be absolutely sure that the samples that they're working with and that data being used is from the right sample and from a quality sample. We employ functional quality control metrics so that we can predict the performance of samples. We make sure that we employ technologies that allow us not only to predict the performance of the sample, but also the sample identity, and then the harmonization of these QC metrics so we can support global collaboration, and doing this real time is critical. So hopefully that sets the tone for the topic for today, and I'm really excited to participate with a number of my colleagues here in digging a little bit deeper into these issues, and then have a question and answer period that can get at your specific questions. So I'd like to turn it over to Faye, who's going to talk about some of the challenges associated with the harmonization of quality control that I just alluded to. Faye?
Okay, thank you, Andy, and hello, everybody from Luxembourg. The question is, why is the quality of biomaterials and all their associated clinical, biological, pathological, pre-analytical data important to biobanks and to the communities that they serve? Now, as everybody knows, there is a kind of continuum in the processes between research and industrial applications, with the output of one process being, of course, the input into another process. And biobanks provide biomaterials and associated data to researchers, so at more than one level in this continuum. Okay. So the first level is when biobanks provide biomaterials to researchers for biomarker discovery. And at that point, accurate sample provenance in terms of clinical endpoint, accurate identity data in terms of demographics, but also accurate pre-analytical data in terms of collection, processing, storage information for the biomaterials are all necessary to support correct experimental planning with comparable pre-analytics. And all this is necessary to have well annotated and fit-for-purpose specimens. Also, proper data management is also needed for that. Now, on a second level, biobanks also provide biomaterials and associated data to industry for biomarker validation purposes. And here, samples of different but always well-documented pre-analytics are needed. We can talk also about the concept of qualified samples when it comes at this level. Biomaterials are also to be used for establishment of reference ranges for new biomarkers. And at this point, many biobanks giving access to specimens from different population groups are needed. And finally, also biological resources, by biological resources, we mean samples plus data, are used for method validation, equipment, performance verification, method comparison, and of course, at this point, we talk about commutability of the biomaterials, which is important, and biomaterials which need to be provided in sufficient numbers and volumes. Now, in order to control and to properly document the chain of custody of the samples and their associated data, there exist quality management systems. And here you have an overview of existing standards. There is a new international accreditation standard, ISO 2387, which has recently been published in August. And compliance to this standard shows consistency and competence of the biobank. There's also a series of European accreditation SEN technical specification standards. You can see some here on the specific area of pre-analytics. More of this is coming. This is an effort where the main driver has been the European consortium SPEDIA, and both clinical laboratories and biobanks are in the scope of these documents. Next, for the documentation and the traceability of the pre-analytics in a standard format, the ISBER has published and twice updated the sample pre-analytical code, the SPREC. And finally, when it comes to laboratory methods, of course, as we know, manufacturer instructions are always to be followed. But also, I would like to add that there's a new standard being developed, a new ISO standard, which contains specifications on the procedures that can be used to properly validate a laboratory processing method. And biobank laboratories are, in principle, processing laboratories, producing samples, molecular derivatives, cellular derivatives as an output. Here we have some more challenges. First, when a biobank is embedded in a clinical diagnostic laboratory, because many biobanks are embedded in clinical laboratories, and the primary focus in this case of the business is, of course, clinical diagnostic assays. So processing methods that are applied for production of the samples, which are in this case the test items, may be considered as simply the pre-analytical phase of a clinical diagnostic assay and not as the main process of interest. There's also the challenge of the interdependency between container, processing, analysis. What I mean is that a specific analysis, a specific assay may be incompatible with a specific processing kit, and the processing kit can be incompatible with a specific tube container. Then we have another challenge relative to exploratory methods, and by exploratory method, I mean a method that the biobank laboratory has never performed before, or a novel method that no one has ever performed before, or an adaptation, modification of a known method. These exploratory methods represent a challenge. We have in-process quality control materials that may be needed to assess consistency in the performance of a processing method. As you can see in the chart on the left side, this is a range chart, and range charts can be used by the processing laboratory to ensure that the average yield, purity, integrity, other quality attributes of the produced material is consistent. We have also external quality assurance programs for processing methods. This is the EQA processing schemes, which again are used to assess the performance of the different processing methods. And they are especially useful when we want to pool resources from different biobanks together, and we want to make sure that quality of the material is harmonized. And in all of these areas, there are challenges relative to the homogeneity and the stability of materials that need to be used, either as an in-process QC material or as an EQA material. There are challenges relative to the nature of these materials. Sometimes surrogate materials need to be used. And to finish, I would like to highlight one issue, which is the standardization in terms of quality control of the different types of specimens. Here, I would like to refer you to this technical report published 2 years ago by the ISBER Biospecimen Science Working Group. And this is essentially a guide to specific assays that can be performed on specific types of specimens in order to qualify them in the scope of a specific use, or when this is not possible, based on current knowledge, in order to stratify the specimens into predefined quality categories. And therefore, the user can make informed decisions on the use of samples and on the best experimental plan. We have also put this guide into the format of a free online application. We call this a Find My Assay. This is, you can click and you can go there. This Find My Assay guide will be updated in 2019. So, to finish, lots of challenges, but also some tools to overcome the challenges in order to integrate the processing and the quality control and to be able to provide samples in which the user can have total confidence. And with this, I would like to pass it to Dr. Shawn at Levy.
Thanks, Faye. My name is Shawn Levy, and as Andy alluded to in the introduction, my laboratory focuses primarily on the application of genomic assays to a wide diversity of samples, both in the clinical as well as preclinical and research space. And I'll take the next few minutes to offer some perspectives that we find more on the laboratory aspects and the integration of and importance of sample quality control as well as sample integrity in that process. So as mentioned in the introduction, I oversee both the research and clinical laboratories at HudsonAlpha, and we're a center that supports a large-scale genomic analysis for the characterization of a number of diseases, particularly looking at both genetic and functional variation in human development. And the ability to analyze and harmonize data, of course, is critical in a number of these efforts. And as shown on the right-hand side of the screen, over the last several years, the routine application of genomics in large-scale studies has become much more common. And so therefore, the control of both batch and temporal effects are important and very important to be minimized, at least as far as the external effects go, because many of these things ultimately define the resolution of the experiments. And as the technologies get better and better and more routine to measure the totality of the DNA or RNA species present, it puts more and more pressure both on the quality of the sample and, of course, highlights the critical importance of the integrity of the sample with respect to contamination and, of course, appropriate sample identification. So not surprising, there's a number of procedures we've implemented, including large-scale automation as well as key sample integrity management at isolation and quality control. In brief summary, essentially all processes involve a scan-in, scan-out process with integrated barcode tracking and validation and the use of what's now become fairly standard in both the biobanking and research space of 2D coded tubes. And then all barcodes, of course, are read and tracked into a laboratory information management system, and automation is used whenever possible. And then we continue to perform isolations on a plate-based protocol with barcode tracking. And what's shown on the left-hand side of the slide is a part of our automated, in this case, stool isolation platform, where it involves the what's not quite visible is there's each of the racks that are holding the taller tubes that you see towards the middle of the slide. As those are entered into the robot, all barcodes are scanned in an automated fashion and tracked. And then as the samples transfer into the processing plates, the 96-well plates you see towards the left of those tubes, of course, those barcodes are maintained within an electronic database and continue to track through the rest of the processes. And then when the tubes are removed, when the racks are taken out, same thing, they're rescanned to, again, verify that no tubes were moved inappropriately during the assay. We do the same thing on all of the downstream assays. Another important factor after the isolation piece, of course, is the continued tracking and integrity measurements. And this is just a very simplistic view of two different assays performed on the same sample. When an aliquot goes off for processing for, in this case, it was whole genome sequencing, a separate aliquot goes off for a fingerprinting assay, and then via our laboratory information management system, the fingerprinting assay is compared to the results from the whole genome assay. Fingerprinting assay has a little over 100 high minor allele frequency single nucleotide polymorphisms, and then those are compared to the final whole genome or whole exome or even RNA sequencing results. And the expectation is that you see an integrity score of 100% or 1.0, as you see in the example samples shown here. So this is a kind of the first step in measuring the integrity of the sample. There's also, because we sequence the fingerprinting assay at very high depth, it also allows a measurement of contamination as well, at least to reasonable degrees. And then finally, as we've moved to more and more automation, we've also started to assess the ability to perform higher resolution quality control, and that involves the actual use of small sequencing or rapid sequencing applications if the endpoint assay is a sequencing procedure, that the intermediate quality control and the detection of molecular identifiers that are introduced early in the sample process, whether they be spike-in identifiers or introduced during the preparatory process, those can be measured and tracked at intermediate quality control steps if those intermediate quality control steps are use a sequencing application, which is kind of shown here in a cartoon format on the left-hand side of the slide, that in this case, some examples of a one whole genome, or even same thing would apply to a whole exome protocol, that as samples go through preparation and then are indexed through some either PCR-free or some low cycles of PCR, we'll then pool the samples and do an initial assessment using an iSeq instrument, low-cost, rapid way to assess 96 to 384 samples at a time. And then from there, be able to both measure sample performance, pooling performance, sample identity, etc., and then can verify at this intermediate step before we move on to larger expense or higher expense assays, that everything is passing quality control, and we even have an opportunity to rebalance pools, for example, if the distribution of the reads per sample is incorrect for what the ultimate assay is needed to be. So even though there's a goal of introducing sample integrity measurements, intermediate quality control measurements, and doing so at the molecular level to enhance resolution, we've also taken an approach to really ask the question, as the technologies get better and the resolution of the technologies get better, what additional optimizations can happen at the sample isolation stage to continue to improve the quality of those samples for the downstream assays. And when I say improve the quality of sample, it means try to get closer and closer to the native state of those samples, in the cellular or tissue from which they are derived. So we've spent some time on that in the last couple of slides. In closing, I'll just show an example. In this case, this is from fixed formalin paraffin-embedded sections. We have similar results from cores, showing just quality improvement in received RNA. So about half of the slide on the left-hand side was a standard protocol where 22 out of 95 samples had a DV200 of greater than 20%, meaning that the average nucleotide length detected by fragment analysis was 200 nucleotides or larger. It was only about a quarter of the samples passed that particular metric. And I think our lab, like many others, has found that samples at DV200 of 20% or higher generally perform reasonably well. And then through an optimized protocol, which we'll be presenting more on and releasing in the coming months, we were able to achieve nearly 100% of samples, achieving the quality value of above 20%. And in fact, as you can see in the graph on the right-hand side, many of the samples now are actually exceptionally higher quality. And this has not been limited to one type of tissue or one tumor type. In fact, this procedure, preceding the optimizations, we had probably isolated 20,000 samples under these methodologies, and we've isolated thousands of samples under the optimized methodology, and this improvement in quality is quite consistent and actually a little bit more striking in the core samples. And what this has the downstream effect on is a bit illustrated here, and that when we look at functional assays, in this case RNA sequencing assays, we can robustly manage the variation we see batch to batch, meaning that the nucleic acid material isolated performs very similarly from one batch to batch number 2. And in this case here, this is about 3,000 samples analyzed under the same protocol. And as you can see in this, or at least hopefully I can convince you that this shows that the principal component analysis showed the batch effect, the actual plate-based batch effect, was in the 5th to 7th principal component, depending on how we which samples we group together. And then what's shown in the black circles in the very middle of the bottom panel is a positive control, or essentially a known control that was included in each batch. So in closing, the importance of quality control and technical process improvement through the totality of the assay, we found that to be critically important in addition to some of the things that have been discussed by the other presenters. So with that, I'll move on to our final presenter, and thanks everyone for their attention.
Thanks, Shawn. As Andy mentioned in the beginning, I'm going to come at this from a slightly different perspective. I'm a co-founder of a company called Life-Alt Bio, which is really delivering a personal biobank to consumers, and we deliver that to consumers off of blood with the idea that we're preserving both the informative as well as restorative potential of blood, ultimately for future diagnostic and therapeutic uses. So at the end of the day, we are in many ways a consumer of the standards that Andy and his group at Brooks established for our processes. So we like to see things at the highest level possible. Being a direct-to-consumer company, we focus on preservation of actual cells, DNA, and blood plasma from a peripheral blood draw. And as you can imagine, there are many uses of the materials that we isolate, both from an analytic perspective of the DNA contained within, but also the many biological and metabolite components and things from the blood plasma, but also the cells for an emerging use of cells in the therapeutic space. With that, we obviously want to maintain and ensure that we have high quality materials preserved for our consumers. And as you can imagine, within this process, there are really 3 points where variability can creep in and inconsistency can creep in, and that's in the collection and the shipment of the blood sample. Likewise, the actual bioprocessing and the manipulation to isolate the materials we want to preserve, and then ultimately in the actual preservation and storage of that. So with that, we look to actually control that in a variety of different ways. Up front on the collection and shipment side, what we integrate to control that process are contracted, validated phlebotomists that ultimately will follow strict protocols in terms of how they collect the blood, into what types of tubes they collect the blood, into how they ship the blood and package the blood. The next step in that process, you imagine we have to get the material actually to the bioprocessing center, but get it in a stable way. So we have qualified and validated the actual shipment protocols that we use, where we utilize FedEx as our primary overnight delivery provider, but we also do that in a temperature regulated manner. And then last and very pertinent to our process is actually having validated and qualified bioprocessing and storage, and that's where we engage with Brooks Life Sciences and RUCDR to provide and implement those standards of practice. As you can imagine, right, consistent process and long-term stable storage are actually very, very essential for the downstream use of the materials that we're storing for our consumers. So if we have low quality, ultimately at the end of the day, the assays or the analytics that people might want to engage, or the therapies they might want to engage with the cells they've stored, they'll have inconsistent results, they'll have poor results, or they'll have unusable cells. But given we're collecting and preserving materials that are going to be applicable in a regenerative medicine or a precision medicine space, we know that sample and process quality equals usability and accessibility. So we need to maximize on our end the compatibility with current and even future assays. So for example, what we currently collect and preserve with our Good Cell product, which is our direct-to-consumer product, is we preserve the cellular potential in the white blood cells that's contained within the blood. We preserve the DNA, which is used in obviously a variety of genetic assays that are available, both direct-to-consumer as well as in the clinical space, and then the blood plasma in the same context. So if we have poor quality supply of the DNA or the blood plasma, we're going to get inconsistent data and things like that. So with that compatibility requirement for our therapeutic processes and our analytic processes, we need to have standardized, consistent processes for what we know today. But what about the samples in the future? We know, for example, we can run genetic assays on the DNA. We can run blood tests on blood plasma like blood cholesterol, blood sugars, a variety of things. But we also know even currently that there are different components in those environments that we'd like to detect. So, for example, the emergence of liquid biopsy or monitoring cell-free DNA. Those are emerging areas where people are understanding and developing the way to properly analyzing and ultimately control the sample for that analysis so you get consistency. Those are being developed. We currently don't integrate those into our process because we don't have a validated method for that. But what about the materials for the future? Well, really it's about, are we collecting and storing the right materials out of the gate? And if we're collecting them, are we collecting them in the best manner? And what's not known is not known. So we can't even store it in the best format. So with that, we need to basically work from an existing platform of standardized process. And that's with what we know. And we know we have validated measures for qualifying the cells that we preserve. We have validated measures for ensuring the integrity and applicability of DNA and downstream assay. And we have validated processes and resources for the blood plasma as well. So really it's about us working from a standard to make changes in the future to evolve with the science that's emerging. And as new scientific discoveries, new analysis and things like that emerge and come online, we will adapt from that platform and make the change from that platform and not from some outside environment. So how do we deliver personal biobanking responsibly to our consumer? At the end of the day, the materials we're storing are for that consumer and for their use. So for us, it's really about meeting the needs and the specs of our partners that we're reporting off of. We are preserving and storing the material and then being the resource of distribution for that downstream analysis or that downstream use, whether it's in therapy or analysis. So with that, what we really look to do is execute with the best partners in the space. We look to execute with certified and accredited labs that are regulated, that utilize standardized processes and methods, highly automated, highly barcoded. We look to standardize materials and use the highest quality materials that meet the highest level of expectation for downstream use. And we look to use partners that have proper management systems in place for us to track and trace the consumers that we bring on board. And lastly, and most responsibly for our consumer, it's really about ensuring that they are provided the most comprehensive, proper consent for, and as alongside awareness for all the potential future uses for the materials that they're storing with us to ultimately minimize delay and utility down the line with providers that are going to use the materials. And with that, I'll pass it back to Andy and transition to the next part of the program.
Great. Thank you, Brad. I'd like to thank Faye and Shawn as well for providing their perspective. And hopefully, for those of you that are listening in, you've got a little insight to some really different areas that require the best in digital and physical sample chain of custody, because as we all know, it really impacts everything we do around the biological sample. So with that said, now I think we will transition into the really the roundtable discussion with some questions from those that are listening in and participating. And now I think you'll receive a few instructions again about how to post questions if you haven't already done so, and then we'll begin.
Thank you again, and ladies and gentlemen, just a quick reminder on how to submit questions. Just click on the Ask a Question box located on the far left of your screen, and we will answer as many of your questions as we have time for. Go ahead, Andy.
Okay, thank you. So the first question I'd like to pose to Shawn based on the question. It's often that you receive samples that come from many different tissue sources and from many different laboratories that ultimately end up, and I'm just paraphrasing the question, that ultimately end up for one analysis or another. How do you really control and or address different tissue sources within analyses, or even different banks or labs that are presenting samples, so that you can be sure that that data is as harmonized or as homogeneous as possible? Or is that not even possible?
Well, I think it is within ranges, and so certainly, probably like many laboratories, we have a series of specifications that we request for different tissue types, and those include, of course, a variety of fixed formalin, paraffin embedded samples, whether they be cores or sections, thickness of section, number of sections, approximate tissue area within those sections, et cetera, as well as the weight of tissues for or cell counts for things like fresh frozen tissue or sorted or otherwise processed cells. And so even within, but of course within those standards, some percentage of samples will meet them. We know how all of the downstream assays perform within those standardized ranges, and so that is one key step for the harmonization. But the reality is that there's always a number of samples that fall outside of those ranges. And when we look at this from a patient perspective, even those samples that fall outside the range, they still represent, of course, critical individuals, whether they be for research or certainly, patients that are looking for answers if they're for clinical. And so we still have to have methodologies that can robustly support things outside of those ranges. And so we kind of call the ranges the ideal, and kind of everything else is still the workable. And we design and have the assay goals. And then, so for things that fall outside those ranges, harmonization is still an important activity. And the reality there, that just really shifts to, instead of looking at purely sample quality control, it becomes a continuum of specifications of sample quality control all the way down through, the actual quality control of the data itself. And then we really just use operationally a series of flags that indicate, where samples pass and where they may fail on those different things. And so depending on the question being asked across these projects, you'd have just different tolerances of flags as far as that, that overall harmonization. So the short answer is there is no short answer, and that and the reality of this space is that all of these samples are precious. They all represent very important research value or clinical value, and so we just do our best to work with them and try to do so in a highly documented and controlled manner.
Are there things that you can learn from an analysis or the QC that you provide on the samples that you get that could be used to help individual sites, investigators, or biobanks that are collecting and managing these samples? Is there information that can flow in the other direction to try and bring that tighter, given that there's only so much that you can do once you get a sample?
Oh, absolutely, and we certainly make those efforts. And what we found in working with, if I kind of take a section of oncology-based projects, which often have the highest number of challenging samples, because the reality is the molecular assays often get kind of the leftovers from the pathology assays. And of course, there's surgical procedures and other things that can potentially compromise the samples further. We've tried to provide as much feedback as possible and have as much integration through the entire sample collection process as possible, meaning that we're not just speaking to the person who ships the samples out of a biobank, but ideally, speaking to the clinical staff, even all the way up through the surgeons, and try to make things as simple as possible and understand their challenges to offer some of those inputs. And so we've tried all of those things, but again, the summary answer is, as much information that can be passed back to the effects of specific sample compromises, whether it be temperature or time or processing, as much clear and distinct comments we can provide, kind of if-then statements back to those individuals. We've actually been able to see, reasonably good performance improvements for a number of challenging sample types.
That's great. Thanks. We have a question that states, or a general question asking, where is the best place to find information about the newest advances in sample quality control, sample chain of custody, and the regulatory oversight of sample collection and processing? Faye, given your experience in the field and your involvement in your role in not only creating these approaches, but educating people and talking to the biobank community as well as the precision medicine community, where do you think the best places to go for people, either basic or advanced, to find where they can get the best information about chain of custody and quality control?
I guess there are not too many places to find the information. Well, when it comes to management of samples, of course, there is the ISPER, International Society for Biological and Environmental Repositories. There are several ISPER-endorsed courses that are delivered across the world. There are also national networks. Some of the national networks are really active and continuously provide information on the website. The US, the NCI, the OBBR program in Germany, the German Biobank Alliance. Spain has a very active network. Canada, the CTRNet network, where people can find updated information. There is also, of course, the journal, if you want to find the up-to-date and latest news on processing methods, quality control, method comparison, and so on, biopreservation and biobanking journal, and of course other scientific journals. And this is, of course, this is another challenge in this community, is that the spread in terms of scientific journals, where one can find the latest scientific information, is so wide. So we once looked across with the ISPER working group, and we could find more than 150 journals where you could find relative information. So I guess it's one who should do PubMed search to be continuously updated, because this area is growing exponentially. When you look for biospecimen research, for example, 10 years ago there was almost nothing, and now it grows exponentially.
Right. Don't you in your role at the IBBL, one of your programs, you run a proficiency testing programs for some processing activities and quality control. Could you maybe share a little bit about that and how that factors into this for those that are asking questions about how they can actively engage in understanding the quality of a sample and their process?
Yes, so we run this biospecimen proficiency testing program for the last 7 years now. So this is an annual program where anybody can enroll online. There is a group of schemes which are processing schemes. So the participant receives a sample, say a stabilized blood sample or an FFPE sample, which has previously, all the aliquots have been tested for homogeneity and stability, right? So the participant performs his routine extraction method, can be DNA, RNA, protein extraction method, can be viable cell isolation, so different methods. And the product, the output, is being sent back to IBBL for isochronous central analysis relative to different quality attributes of all these samples that have been extracted or isolated from each laboratory. And then there's statistical analysis, so each laboratory can see and benchmark the performance of its processing method relative to all other 50 or 60 or whatever laboratories across the world. So this is the processing schemes, and there is another group of analytical schemes where the focus is analytical methods, and these are now, we're not talking about clinical diagnostic methods, we are talking strictly about analytical methods which are performed to characterize and quality control different types of biospecimens. So there are schemes for DNA quantification, RNA integrity, cell viability, tissue histology, and so on. And so in this case, the participant receives, again, a standard item, it's like a reference material with an assigned value, reference value. Everybody does the measurements, submits online the results, and then again you can see what is the accuracy and the precision of your analytical method. So, and
great,
of course,
thank you.
Yeah, it's a kind of education also, because in case of discrepant results, there is feedback and advice, and so on.
Yeah. So thank you, Faye. The next question relates to sample quality control and data quality control in the direct-to-consumer applications, or in the role of personal biobanking. So maybe Brad, you can field this one, is what are some of the challenges that you face in identifying quality metrics for samples that you describe were being collected that you don't know what those downstream applications are? So how do you best prepare for analyses and or cellular therapies, I guess, in the case of LifeVault? How do you best plan for that? And how do you qualify those samples? And is that a question that we can answer even now?
Yeah, I mean, a good question in itself, right? With the emergence of cellular therapy in a variety of different formats, whether it's blood stem cell transplant, whether it's CAR T therapies, or the emergence of, iPS-derived differentiated cell product therapies, developing the standards of what a qualified material is at the endpoint is still being resolved. And I think is a little more resolved on the upfront, because what we're really storing is the material to derivatize off of. So we store, the way we kind of preserve potential and try and address the challenge of what we don't know is we try and sort of minimize the process with what we do preserve. So with what we break down, we actually preserve the PBMC layer, which actually houses 3 of the most commonly utilized cell sources for cell therapies right now, right? So it carries hematopoietic progenitors that have been used in blood stem cell transplant for decades. It carries the T cells that are being engineered and reinfused into patients to cure different lymphomas and leukemias. And it also carries the substrates of cells that'll be utilized to derive induced pluripotent stem cells, which are the substrate for personalized stem cell therapies. So there are ways that we can measure and monitor those. So, some of the basic metrics that we apply are simple things, right? Can we secure the right identity of the cell to the best known standard that is published? Particular marks, for example, for hematopoietic progenitors, what's the viable CD34 cell population. For T cells, we can utilize a CD3 mark or more specific subset marks. For IPS, we actually go and we validate the ability to actually isolate the primary cell type that is going to be the substrate for the reprogramming process required to make these IPS cells. So we qualify the process to basically establish that. We then, of course, quantify through different marks the presence of that particular cell type, and then we validate the ability to make the cell. But at the end of the day, the utility of the materials that we're isolating that are known quantities, their entry into therapy is going to be defined and specified by the companies and the institutions that are developing those treatments. The unknown ones, will continue to have always the same challenges: the identity, the purity, and the potency.
That's great. Thank you, Brad. We have time for one last question. With respect to transcriptomic samples or transcript profiling, there's a question about spike-ins and other controls. Shawn, maybe you can address this. The question with respect to transcript profiling and metagenomic analysis, so whether it be fecal samples or RNA from blood, saliva, what have you, tissue, what controls do you recommend so that you can assess both sample quality but also performance of the assay, and are spike-ins used regularly?
For some designs, spike-ins are used regularly. It really depends on the overall goals. And so 2 simple examples would be a transcriptional profile in, say, a tumor versus normal tissue, which, with the goal of identifying transcripts that either have functional significance or have an ability to distinguish, or transcripts that could be used for analyzing the functional consequences of particular mutations that may be driving the cancer, or even used in assessment of therapy or residual disease monitoring, et cetera. Another example would be, say, on the metabolic side or microbiome side, looking for transcripts that either indicate the functional state of the different species of bacteria that are present. And so in each case, they're generally rooted in counting applications where you're trying to determine the relative amount of any given transcript that's present in comparison to the relative amounts of other transcripts, either from the same species or different species, depending on which one of those applications. Now, the spike-ins can be very helpful, I think, in two ways. One is they can be used for direct sample integrity measurements, and that is where you just have a simple synthetic library molecule that's spiked in, where the segment, the insert on that library molecule is unique to a given sample. But that spike-in alone is great for sample integrity measurements and can be spiked in at very, very tiny amounts in the preparative process because you won't need a lot of reads to detect them on the downstream side, and certainly don't want to consume a lot of resources from your assay in testing those. But it doesn't have any measurement effect in terms of dynamic range or overall measurements of consistency in the assay. And so then two other spike-in opportunities are available. One would be detected during the quality control process, where you have a probe-based assay that can be detected during the real-time quantitation of the overall libraries. Those, I think, are not well used because of the complexities involved and the variability in their performance, at least in our observations. And so the other spike-in, which is probably more valuable and more generalizable, is where you spike in a known collection of transcripts. In this case, you could use them in combination with a sample identity spike, but they could also be used completely independently, where instead of having a unique insert per sample, instead you have a dynamic range of transcripts spiked in at different levels, where you can then assess within a particular assay what is your lower limit of detection, your upper limit of detection, and then what is the linearity of that detection in the overall assay. And then how those spikes are used. So, for example, at the prepared library stage would mimic or allow you to measure the downstream sequencing process. If they were spiked in as synthetic transcripts, which again are a little more expensive, a little harder to control, but at least go through the entire preparatory process, have significant value as well. So, at the end of the day, setting up the spiking controls can be complicated, and I think needs to be integrated into the overall experimental design to be sure that the appropriate measurements and value will be derived from them. And then there's always, of course, the ongoing overhead of those spikes, so they have to be continually evaluated to be sure that they're giving an accurate representation of the assay. But the latter example that I gave, I think, is probably the most valuable and can be most helpful in supporting assays that have very wide ranges of detection needs.
Got it. Thank you. We do have time for one more question, so I'm going to grab another question from the list. And this question is about nucleic acid extraction, DNA and RNA extraction. And the question is, are there methods that are standardized across sample types? And I'll address this question or take a stab at it. The short answer is yes. There are methods that are standardized for DNA and RNA extraction across sample types, and it really requires an evaluation for the lab that's performing this to understand what their metrics are for success. And in fact, in my opinion, I think that there's still a utility for using the 4 different modalities for nucleic acid extraction that exist, and that would be either organic, salting out, solid phase extraction, or bead-based extraction. And given your downstream analysis and what you're looking for in your sample quality, you may choose one or the other and then standardize across tissue types or sample types. So, for example, for investigators or people that are interested in assays that require very high molecular weight DNA, salting out is still the best approach. And if you're looking for those kinds of those assays, that's what you would standardize against for your extraction. When you're looking at limiting sample amounts, people still work with more organic approaches that may give you a little more of a dirty sample, but using the right QC approaches, you can predict the performance of that assay. So in contrast to someone that's doing telomeric length assays and salting out, to someone that's working with very, very small amounts of tissue and just wants to get the most out they can, they may have a different approach. Where I see the field gravitating towards are bead-based approaches in both research applications as well as in clinical applications. And that gives you the widest utility of extraction approaches across tissue types. So if you can use the same chemistry and modify the binding conditions, certainly your elution conditions based on the amount of sample that you have. This gives you the broadest success. Those approaches are also amenable to being nucleic acid agnostic, if you will. So you could look at DNA and or RNA. I'm not implying that it's a co-extraction approach, but being able to use the same chemistry and automation gives you that precision. And at the end of the day, you have to have an analytical and functional quality control assessment of your nucleic acid to make sure that approach works for your downstream application. So it's not just about the yield and the purity, but it's about the correctness of the sample, making sure that you test, as Shawn had pointed out, for sample contamination as best you can. And those are the things in combination will lead to the best standard approach to nucleic acid extraction.
Thank you again, Andrew Brooks, Faye Betsu, Shawn Levy, and Brad Hamilton for your time today and your important research. We would also like to thank LabRoots and our sponsor, Brooks Life Sciences, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information that you provided at the time of registration. This webcast can be viewed on demand through August of 2019. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. A poll will now pop up on your screen. We thank you in advance for participating. Until next time, goodbye.